This is our presentation of gene expression, how hepatitis C can cause insulin resistance, by Gemma and Gemma. The central dogma of molecular biology describes gene expression from DNA to proteins. DNA is a central source of genetic information. It passes on this information to form RNA, which passes on this information to proteins. DNA is able to pass its information on to later generations by replication and to RNA through transcription. RNA then finishes the process by forming the required proteins through translation. Replication is the process that produces new copies of DNA molecules. A gene is a portion of a DNA molecule that carries specific information. The portion of the gene that codes for specific information is the exon. The intron doesn't code for specific information. The DNA molecule unwinds or unzips and opens to produce two exact copies of the DNA molecules. New nucleotides bind to the backbone of each strand of the open DNA by forming hydrogen bonds to the nucleotides already present. This process continues along the open DNA strands until each half of the original DNA has a complementary strand bonded to it. This produces two DNA double helixes, each with half old and half new DNA.
Okay, so transcription is quite a large topic, so I'll just be briefly outlining the main features of the process in order to explain how hepatitis C could cause insulin resistance. Simply put, transcription is the synthesis of RNA from a DNA template and has three main parts to it, regardless of whether it is transcription of prokaryotes or eukaryotic DNA. The first stage of transcription is the initiation, in other words, when RNA polymerase binds to the DNA at a sequence called the promoter. The second stage, elongation, where the nucleotides are added to the three end of the polynucleotide chain. This involves the development of a single strand stretch of DNA. The final stage is termination. This is where the RNA polymerase recognizes the transcription termination sequence and is released from the DNA template. Only selected genes are copied. The synthesis of RNA is somewhat like the synthesis of DNA in that they are both synthesized in a 5-to-3 direction. However, RNA polymerase does not require a primer and lacks the ability found in DNA polymerase to excise mismatched nucleotides.
Translation is the synthesis of proteins. It occurs in the ribosomes. The ribosomes are complex units made of RNA and protein. Different subunits of rRNA hold on to mRNA in place during translation. The mRNA resulting from transcription gains a cap and a poly A tail before it travels out of the nucleus to the ribosome. The base sequence of the mRNA is read in a 5' to 3' direction. tRNA contains a complementary sequence to the codon called an anticodon. tRNA brings the correct amino acids into the ribosomes by Watson-Cricket base pair in its anticodon. The ribosomes attaches amino acids to the growing polypeptide chain.
Chronic hepatitis C can induce both insulin resistance and type 2 diabetes in vitro as well as in vivo. This is because both the structural and non-structural proteins of the hepatitis C virus, also known as HCV, hampers insulin signalling by modulating cellular gene expression. This could lead to type 2 diabetes if the level of insulin resistance is significant. Any change to the signalling of glucose uptake is likely to induce insulin resistance. Insulin resistance is a condition where the body is unable to maintain homeostasis, in this case the maintenance of either glucose levels in the body or normal insulin concentration. HCV infects the hepatocytes, which is a functional cell in the liver which carries out metabolic, endocrine and secretory functions, which eventually leads to insulin resistance and type 2 diabetes. Type 2 diabetes prevails in 24 to 50% of chronic HCV patients. The genome of HCV gets translated into 10 different proteins. It is these proteins which help the virus to replicate and damage the host. HCV proteins, three main ones, are core, N53 and N55. are mainly involved. HCV core protein is pathogenic and induces several metabolic disorders in the host cell, including inducing insulin resistance by the way of core protein induction. N5-3 protein's direct role in inducing insulin resistance is yet to be determined, but it does induce oxidative stress in the cell. N5-5A protein interacts with PKR, which is an RNA-dependent kinase. It's located on the ER membrane and affects the modulation of intracellular signaling involving transcriptional factors. It also causes damage to the hepatocytes, which induces ER stress, which then leads on to cause insulin resistance. The infection promotes the expression of gluconeogenic genes, which enhances insulin resistance also. In conclusion, the HCV uses different proteins which can cause insulin resistance. Yeah, and there's the references for the PubMed that we got the information from.
Thank you for listening to our presentation.
Goodbye.